All it aspirants, you are listening to podcast Botany for NEET by S. Ramakrishna. And in this episode, the photosynthesis topic, early experiments. The gradual understanding of photosynthesis simply attributable to contribution of the scientists in earlier periods. Joseph Priestley, Ingenhousz, Julius Sachs, Engelmann, Cornelius Varney. First, Joseph Priestley. Discovery of oxygen attributable to Joseph Priestley. And Joseph Priestley conducted an experiment, role of air in growth of green plants. What is the role of air in growth of green plants? Plant requires air. It takes carbon dioxide. It gives out the oxygen. That means it not only takes the carbon dioxide, by giving oxygen restores qualities the air. And in this experiment, burning candle placed in a closed space, that is in bell jar. Soon the candle went out, extinguished. Who placed a mouse would suffocate. The atmosphere inside the bell jar fouled, contaminated by burning candle or breathed by animal. Who placed a mint plant, the mouse stays alive, candle continues to burn. To burn the candle, air is required. To breathe by animal, the air is required. So plant restores the air earlier fouled by burning candle and breathed by animal. Next, how Joseph Priestley would have conducted the experiment with a plant and a candle, and he would rekindle the wick of the candle. He would rekindle means that is relit the wick of the candle, whether the candle continues to burn after some time. How many ways you might have used to relit the wick of the candle without disturbing the setup? We may think you might have used a converging lens, or you might have used electric spark to lit the wick of the candle. Next, Ingenhousz, who continued the experiment of Joseph Priestley, once in light, once in dark. According to him, the role of light in purification process by green plant. So green plant purifies the air fouled by burning candle and breathed by animal only in the presence of light. And who conducted an elegant experiment, easy to understand, that is hydrilla funnel experiment. This hydrilla plant is a submerged suspended hydrophyte. In presence of bright sunlight, green pods could release bubbles of gas, nothing but oxygen. It releases the bubbles of gas only in the presence of light but not in dark. Next, Julius Sachs. During the growth of plant, glucose is made, the glucose stored as the starch. And the green pigments, now we are calling it as the chlorophylls, located in special bodies, now we are calling it as chloroplast. So pigments, the green pigments located in chloroplast. And according to him, the starch is a storable form. Next, Engelmann. We have to remember the key points in case of Engelmann. Cladophora. It is green, multicellular, unbranched, filamentous alga that belongs to chlorophyll. And aerobic bacteria who uses prism. So, Cladophora illuminated in presence of the light, suspended with aerobic bacteria, split light through spectral component. The aerobic bacteria collected at blue and red part of the spectrum. So at blue and red part of the spectrum, oxygen evolved. So there is a survival of aerobic bacteria collected in blue and red part of the spectrum. So photosynthetic yield in terms of what in this experiment? Oxygen. So first action spectrum, that is graphic representation of photosynthetic yield in terms of oxygen attributable to Engelmann. Next. And understanding of photosynthesis, better understanding of photosynthesis just in the middle of 19th century. And a milestone contribution, a great contribution by microbiologist Cornelius Van Neel, who conducted an experiments in bacteria, purple and green sulfur bacteria. Next. Whatever the hydrogens required for reduction of the carbon dioxide are available from water. It is an oxidizable compound. So hydrogens required for reduction of carbon dioxide available from oxidizable compound. What is oxidizable compound in plants? Water. What is oxidizable compound in case of the bacteria? It is H2S. A simple equation CO2 plus H2O. CH2O plus oxygen. And in case of the Cornelius Van Neel equation, 2H2A plus CO2. 2A plus CH2O plus H2O. To A substituted with it is oxygen. That is 2H2O plus CO2, oxygen plus H2O plus CH2O. In case of the bacteria, 2H2S plus CO2 gives rise to 2S, the sulfur, plus CH2O plus water. So hydrogen supplied by H2S in case of bacterial photosynthesis and hydrogen supplied by water in case of plant photosynthesis. Next. And according to Cornelius Van Neel, photosynthesis is essentially a light dependent process and oxygen evolved from water, not from carbon dioxide. But oxygen evolved from water, not from the carbon dioxide, not experimentally confirmed by Cornelius Van Neel. Later, some other scientists contributed to prove that oxygen evolved by splitting of water by using a technique, radioisotopic technique. They not use radioisotope. They use a radioisotopic technique, but they used heavy isotopic oxygen, that is O18. So oxygen evolved from splitting of water. So in that equation, 6CO2 plus 12H2O gives rise to C6H12O6 plus 6H2O plus 6O2. So 6O2, 12 oxygens coming from the 12 molecules of water. Next. So in this photosynthesis, why 12 water molecules are utilized? As C6H12O6, in case of the glucose, it requires 12 hydrogens. That means for reduction of the carbon dioxide, 6 molecules of the carbon dioxide, 12 water molecule has to be oxidized to reduce 6 molecules of carbon dioxide into C6H12O6. I am signing off from this episode. In next episode, we will see where do photosynthesis occur. So listen with concentration, listen with attention, connect to the concept and recall the answers without any confusion at the time of examination. And if you feel this approach is right, subscribe and share to the person, those who are seriously preparing for NEET.